So it is 12 noon in Los Angeles, and welcome to our end of the year Enigma teleconference, hopefully the last teleconference of the year. And again, very, very appreciative to everyone joining us today in a very busy time, particularly in time zones that are less easy to join than it is here in Los Angeles. So just a quick shout out to the folks here at USC. You can see them listed here. We couldn't certainly do any of this without the support of the folks listed here. So we really appreciate everything that you've done this year. So here's a summary of today's call. We're going to try and get through this in an hour. There's 170 slides, so that should be a piece of cake. It's going to be a year in review. It's starting to get where there's so many productive things and achievements and celebrations that we can't really fit it all in. So again, I apologize if I don't highlight things that some things that were important, but we'll try and give a digest that is hopefully helpful. So first of all, Enigma's 15. It's Enigma's 15th birthday, or it was a couple of weeks ago. We'll show you some highlights from this year's conferences worldwide, SOBP, OHBM, the summit in Amsterdam a couple of weeks ago with 100 attendees. We'll give you an overview of the grants that are active, new, pending, and planned, a lot of publications this year. And then I'll try and give you an overview and a little bit of a summary of the genetic and epigenetic work, the clinical highlights, new methods, new workflows you might want to try, and then also some new working groups and novel initiatives you can take part in. And there'll be a group photo. If you're in Eastern Europe and want to go to bed, that's fine. You can just send us a picture of yourselves. But if you're still on the call at the very end, we'll have a group picture. So Enigma's 15. I want to just say a huge thank you for everything that you've all contributed to make this a tremendous journey. Professor Odile van den Heuvel hosted us in Amsterdam 2 weeks ago, and it was part of a tremendous workshop that was a day and a half. And thanks for being such great hosts. You can see Odile, Chris, Naja, others from the Amsterdam group here. And we even had a cake, so very, very, very kind. So thanks for making the Enigma's now a teenager, so thanks for making the birthday a memorable occasion. Just to sort of remind people, so 100 years ago, or actually 8 years ago, this was some of the Enigma chairs in Atlanta at SOBP 2016. And I think all of you are still here. I know Dick has retired, but you're on the call. Thank you. And I think everyone is still with us. So just some highlights of the year in May at SOBP. A lot of work was presented, some great symposia, nice social event here. You can see many of you presenting or taking part in different activities here. It's kind of a cool group photo of an event that we had there in Austin in the evening. And then also at OHBM, I want to thank particularly Sophia and also Gaon Kim, who's a graduate student here who grew up in Korea, for finding a good venue. This was a wonderful rooftop venue to have a social event. And I want to thank Andrea Arimia. This is Andrea Arimia's drone video of the event. And thanks, Andrea, for smuggling a drone into Korea and being able to do this. I'm sure it's legal. So anyway, just some other highlights of folks presenting. Thanks to all of you who presented at SIRS, International Society of Bipolar Disorder, sleep conferences, ICP in Antalya, Turkey. You can see on the bottom right our colleagues at the SOCAT lab and at Brigham in Antalya, and also the American Epilepsy Society down on the bottom left. There was a terrific gathering a week ago when we were in Amsterdam here in Los Angeles. Neurotrauma, you can see Dr. Carrie Asapenko winning an award for her brain trauma work in the middle row, second picture in. And also a lot of great things being presented at SSSP. EMHFC is a fear conditioning meeting, so you can see Enric Vilahosana from the IMARD group in Barcelona presenting, and I'll tell you about their work in a minute. Lots of good exchanges. Thanks for coming to Los Angeles. We had a visit from Yidin Gao from the Antisocial Behavior Conduct Disorder Working Group, Han Wen Bi from the Forschungszentrum in Jülich, Willem Bruin, it was great to see you, Stefan Ehrlich, Yuji Zhao, and Willem again giving a talk, Willem from Leiden in the Netherlands. I mean, we learned a lot from you. Anyone who wants to come see us in Los Angeles, you're very, very welcome. I think that's been one of the most rewarding aspects to have some of the trainees do some exchange visits. So just as we said earlier, stay on the line. We'll have a good group picture at the very end, hopefully in about 50 minutes. So here's a map. You all know this. This is the current map, I think. If you're somewhere that's not on it, send Sophia a note. But the key at the bottom are the 50 working groups of ENIGMA, the different clinical and methods working groups are shown in the key, and the various locations of the scientists. Do email Sophia if you're not on the map. So here's a current summary. The clinical and technical working groups of ENIGMA are organized into those focusing on psychiatry, neurology, brain development, some on infectious disease and neurogenetics. You'll see some new ones over on the far right, which I'll tell you about at the end, and then some methods and technical groups up there in black and white at the top. Just to sort of remind folks, many of these groups now have subgroups. So the anxiety working group over on the upper left has specialized groups focusing on panic disorder, specific phobia, fear conditioning, and so forth. And Emily Dennis, Lisa Wild, and David Tate's brain injury working group has many subgroups looking at sports injury, military brain injury, intimate partner violence, and so forth. So we'll give you a quick roundup of what the groups have been up to. So we had 2 weeks ago a tremendous gathering in Amsterdam. This began as a neurodevelopmental working group meeting and expanded quite a bit. A big thank you to Nadia Odile, Chris Friend in Amsterdam, and the ENIGMA PD group, who you see pictured there. It grew from about 20 or 30 to about 100 people, and it was really, really appreciated getting together. There's a beautiful picture of Amsterdam from the canals. Now, just for those who couldn't make it, the reason to do it was the following. So a lot of the neurodevelopmental working groups have become very successful. So there's new grants for groups focusing on anorexia, Tourette, OCD, Origins as an infant development group led by Rebecca Nickmeyer. Many cross-disorder papers. The Antisocial Behavior and Conduct Disorder Working Group has been prolifically publishing many new results on autism, ADHD, binge eating, which we'll look at in a minute. But all of the lead analysts had not met in person. And so given the potential for cross-disorder connections and joint projects and even not reinventing the wheel, we got together. We did also discuss some workflows that we could align across ENIGMA, particularly for resting state, functional MRI, and thanks to everyone who showcased their methods that all the groups could try. So obviously, functional MRI and diffusion MRI, everyone is trying, but there were also methods for tractometry, VBM, cerebellum, and we'll look at those in a minute. So some of the common themes in the last year were a huge surge in the number of groups publishing functional MRI studies. We'll summarize work by Clara, Ilya, Leah, and Frank on the protocols for that. Normative modeling with Riyang Gi, Sophia Frangu publishing a really landmark paper in Lancet Digital Health on normative charts for the lifespan. And then Neda and her DTI team publishing a lot of methods to harmonize DTI-derived measures from multiple cohorts across the lifespan, and we'll highlight some of the tools they've been developing in a minute. One key development was the creation of a consensus workflow for functional MRI analysis to be used in ENIGMA. So it's a big thank you, obviously, to the folks on the right who are the protagonists, and to Tomasz Fodran, Violeta Gispedes Izquierdo, and Nadia Ginalia for their work on this. Just to explain this, 18 ENIGMA groups are analyzing resting state fMRI. Some have published papers, others are processing it, and others are planning an analysis. And the majority are using the half-pipe workflow, which is based on fMRI prep. But the settings were slightly different. So the ways people were processing the data was very, very subtly different. And you could say, well, does it really matter? And Melody Kang here at USC, working with Chris Ching, also at USC, found that if you ran even half-pipe with the same pre-processing, but you differed in whether you applied various settings such as ICA-Aroma, CompCore, global signal regression. The results were really completely different, and you can look at her abstract that's been done with a bipolar working group. There's plenty to say, but suffice it to say at the moment that obviously, it matters what settings you pick. So Clara Moreau, Ilya Veer, Leah Waller, and the Charité group surveyed, I think, around 30 ENIGMA groups about what settings they would like to use. So which ones were important to harmonize, the most important being confound removal, motion scrubbing, and global signal regression, and also which settings would make the most difference to the results. And so that led to a consensus workflow that we'd really like to request people to try. So the goal isn't to stifle creativity. If someone has a different idea, that's perfectly fine. But if you could request the people proposing analyses to use at least this, it has 3 parallel workflows. It will do atlas-based connectivity, seed-based, and network template regression with Vince Calhoun's NeuroMark. And there'll be some standard criteria for QC and motion correction. This was helped a great deal by earlier work by Karen Kayenbergs and Frank Hillary with a traumatic brain injury group, where they published ENIGMA's Simple 7. So it's 7 recommendations for functional MRI processing that will help to get reproducible results. So some of the imminent updates in half-pipe, and again, thanks to Tomasz and Violetta and Ilya and Leah, is to upgrade the version used from fMRI prep version, the version that was being used in 2022 to the current one, improve consistency checks, comparing whether a change in the software affects the results versus previous results, and a number of other things that will make this much easier to use. And also there's been some requests to implement motion scrubbing, template-based ICA, surface-based fMRI analysis, and also compatibility with CoinStack, which is a federated analysis framework developed by Vince Calhoun's group. So to really try and coordinate this and allow people to help each other, Clara and Ilya suggested having a regular call. And so if you have people in your team analyzing resting state fMRI, just add yourself to the mailing list. You don't have to join every call, but it'll be a group of people that can share experiences in running analyses. So just to show you some results, Delin Sun had a really nice study of resting state in PTSD across 34 study sites with really robust differences in some of these voxel-based measures. So ELF, ELF, and regional homogeneity, the measures that are associated to some degree with perfusion. And they were finding group differences in some of the regions implicated in PTSD. Another way to look at this, also from the PTSD group, was led by Carissa Weiss-Thomas. She found that the dynamic profile of connectivity did not settle down as quickly in PTSD as it did in controls. So that adds a new perspective to the sorts of things that might help detect a diagnostic difference. This is a very cool study. So Leah Waller, who is the mastermind with Ilya of Half-Pipe, is finishing her thesis doing a genetic study of task-based fMRI. This is phenomenal work looking at 4 tasks, showing that the activations are heritable, even finding single nucleotide variants in the genome that are associated with task response. So it's extraordinary, I think, Leah, that you are only just finishing your PhD, but you contributed so much to the success of all of these resting state studies. This is really cool work, task-based fMRI in OCD, led by Nadia Ginalia, looking at emotional domain and executive domain tasks. Many manuscripts underway, and Chris Friend sent me an email this morning that Nadia's paper was just accepted. See you in 2.5 hours. So very, very hot off the press. Congrats for that paper getting in. Miguel Angel Fulana Rivas in Barcelona and his colleagues integrated task-based fMRI with a fear conditioning paradigm from huge number of sites, over 40 sites looking at brain activation during fear conditioning, showing that it was very different in those with anxiety-related and depressive disorders, both at the group level and in the frequency of individual differences that they identified with normative models. Quick summary of grants. So, congrats to everyone who's won funding to do multi-site work with Enigma. The top one there, Hugh Garavan and his team renewed their addiction R01. I won't go through them all. We're just very appreciative of all the time people put into these. Just as a summary, many of these in the United States are funded through NIMH, NINDS, NIA, but there are a couple of new ones. There's one from NIHS to Lauren Salmon and colleagues to look at environmental effects, and also NIDDK to Shan Luo to look at diabetes. So it's really exciting to see. Just awarded, congrats to Theo, Gemma, Andre, and Jess for their schizophrenia R01. A great one on tractometry. Congrats to Neda, Yisbrand, and team. A new one to collect data in Pakistan. So Maheen Adamson and Mohammed Parvaz with their colleagues at AKU in Karachi are going to be collecting lifespan data there. New grants on anorexia, EEG to Dirk Smith, bipolar disorder. Chris Ching and Matt Kempton have won a grant to run VBM. And also, we may get this before the year is out. CentileBrain, which is developed by Sophia Frangu and Riyang Gi, may be awarded a grant very soon. OCD on the third try was awarded, and a K award to Kathy Lawrence to study autism and ADHD was just awarded, and also a NARSAD award to Leila Nabalsi. Congrats to Leila for her work on bipolar being funded. These are all going back in. I won't read them all, but congrats particularly to Yana Marie for being invited to put in a full application for her anxiety-related grant. Two really cool capacity building grants. So one in India, led by the group at NIMHANS in Bangalore, is collecting brain imaging data they've scanned now over 400 people. And just a few weeks ago, Maheen Adamson and Mohammed Parvaz were funded to collect similar data in Pakistan, in Karachi. So we wish them the very best in collecting that data. It will be a very valuable addition to the diversity of the cohorts that we're assessing. I would add also that Jahanshad has a grant on MENA, Middle East and North African ancestry people, that was recently funded, which will also add to the diversity of the cohorts. This is the India group. Tamagna Chattopadhyay going to visit them and talking about deep learning. Very, very strong group in neuroimaging that's contributed extensively to Enigma, including the OCD group and many others. So thanks for your contributions. And that's how it is in Bangalore. Beautiful there, if you ever get the chance to go. Publications. Huge congrats to everybody who's published Enigma-related work this year. I won't have the chance to read them all out, but you can see a full list. Sophia has put links in. So what will be rather nice is we'll send this Google slide deck out afterwards. If you'd like to browse these and have a convenient place to find the papers, this is it. I'll focus on a couple of these papers that might be of general interest today. There's many more papers that are submitted. You may see some papers here that you have contributed to, and thanks again to all the authors for your perseverance and success in getting these in. These are mainly preprints, so there's quite a lot of these that are under review. Again, do take the time to browse these using the links that Sophia has put in at the end. Some of the methods highlight. So let's talk about some of the new methods and new projects. So you all know this. I mean, those of you that lead analysis or chair a working group or contribute to analysis know that at the top, there's primary workflows to analyze structural MRI, diffusion MRI, and now resting state and task-based fMRI. There's also, led by Alison Nugent, a terrific workflow to analyze MEG. Dirk Smith and Philip Jawinski have been running EEG protocols for quite some time. But then there are a lot of other methods. So what did you sort of tell people about how good these are? You know, the Cerebellum pipeline, the VBM pipeline, and CAT12 being used a lot. So I'll just show you some examples of them being used. And then to some degree, these other methods have been used in working groups to look at structural covariance, normative models, gyrification, and so on. So we'll have a quick look at what is going on there. So one of the major papers of the year came out in Nature Genetics. And so Luis Garcia Marin, Adrian Campos, Miguel Renteria, with help from Sarah Medlin's group in Brisbane, and a vast cast of people that were collecting genomic and imaging data. They found over 500 genomic loci associated with the volumes of subcortical structures. So this is, I think, the largest imaging genetic study I've ever seen, and to some extent vindicates the idea of using images to search for genomic loci. One reason that this is interesting to people in psychiatry and neurology is that a lot of the loci that influence or are associated with brain metrics are also among the risk or susceptibility loci for disease. You can see some commonality between the Parkinson's and ADHD risk loci and the ones that affect the brain, giving you a clue about what they're affecting in the brain potentially. And this is kind of extraordinary. So you can predict regional brain volume, or you can account for up to 10% of the variance in these structure volumes just from SNPs, just from SNPs that were discovered in this study, which is varies by ancestry, but is actually a phenomenal result that genomic data accounts for so much of the variability. So I think that truly vindicates the program of doing imaging genetics, and you know is showing us the power of it. A lot of good media coverage. I mean, it's really nice to see this getting some visibility. And again, congrats to Miguel and your team for getting this to the finish line. It was also a collaboration with the Charge Consortium and leveraged the UK Biobank. So we want to acknowledge the people that created those data sets as well. This pipeline is extraordinarily useful. It's been created by Rebecca Carestes, Ian Harding, and used in many settings, including this one, which is a GWAS. It's still open if you want to join in. They have a phenomenal number of cohorts already. The loci they've discovered as being associated with overall and regional cerebellar volumes have a strong overlap with genomic loci implicated in Parkinson's, education attainment. You can see in red on that plot in the middle, there's a lot of things coming out. If you look at pathway analysis, genes expressed in the cerebellum are among the top enrichment categories, which is very encouraging. So if you want to join in, there's still time. I think if you do it in the next month, you'll be in the discovery cohort, and if you need more time, you'll be in a replication cohort. So congrats to Ian and Sarah and all the people contributing. Vilta Baltramaitite, Esther Walton, and Denai Dima are completing a brain age GWAS. So brain age is how much older your brain appears to be than your chronological age, and you can subject that to genetic analysis. So they are still open for people interested in doing this. They have some genome-wide significant hits that are very interesting now that they have nearly 40,000 people in it. And these markers are interesting. They're linked to type 2 diabetes to some extent with educational attainment, so they make a whole lot of sense. So do join in with their project if you are interested. Still waiting for 10 cohorts to run the GWAS. Now this is extraordinary. Dirk Smid and Philip Jowinski have finished a GWAS, a search of the genome for loci that are associated with various metrics from EEG. And so you all know that the coherence of EEG signals and also other measures of connectivity can be calculated. They have a huge number of genetic discoveries that overlap with known genomic markers that are implicated in epilepsy, which is very sensible result given the relevance of EEG to the study of epilepsy. And I just want to say they were just funded by the NWO. Congrats to Dirk and colleagues for winning a major grant in this area. So if you have EEG from clinical cohorts, not necessarily with genetics, you don't need genetics, do reach out to them because they're trying to see what sorts of analyses might be interesting, maybe a normative model or studies of different disorders if there's sufficient interest. This work by Rebecca Nickmeyer's Origins group studies the genomic loci and other factors that affect infant brain development. They've been studying 30 cohorts with structural and DTI resting state data from a little over 4,000 children. And they've been looking at GWAS again, genomic search for loci that affect functional activity, CSF metrics, a measure of immune function, complement 4 variation, and also rare variants, deletions and duplications that might matter for infant brain development. This brilliant paper, which actually was from the end of last year by Alex in Nature Neuroscience, showed that preterm children catch up in their brain growth, at least to some degree, with non-preterm kids. And there's cool new work looking at maternal depression and maternal obesity to see if they modulate or influence these growth profiles. Very, very exciting work being done by the Origins group. They also are looking at resting state networks. So something quite interesting, led by Wei Gao at Cedars-Sinai in Los Angeles, is whether or not you can find these resting state networks in infants. And the picture on the right shows that you can, at least at 1 year of age, beginning with the primary sensory and auditory visual networks, and then moving on to the salience networks later on. So there's an ongoing program that Wei and others are trying to figure out about how you would bridge infant data with resting state data from the other ENIGMA groups that study children. This is exciting. So Sylvain de Rivière and colleagues at King's College London searched the methylome. And so you all know that the genome is methylated. These are epigenetic changes. And the methylation of homeobox genes was found to relate to brain structure. It's pretty exciting. So they searched the genome for places, CPG sites, where the degree of methylation was statistically linked to measures from the MRIs. Homeobox genes, if you remember your developmental neurogenetics courses, are very well known. They encode transcription factors, and they specify the identity of different body segments, ensuring the correct structures form in the right places. So that's a very, very exciting story with genes implicated where their function is very well understood. And there's the Epigenetics group in Amsterdam 2 weeks ago. So thanks to you for your great work on this. Some clinical working group updates. So we'll have a quick look through this. So this is work by Yidian Gao, Marlene Staginis, Stefan de Brito, and Graham Fairchild, looking at antisocial behavior and conduct disorder. So they found among the most profound differences we've seen in developmental neuroscience in regional cortical surface area in people with ASB and conduct disorder. The effect sizes around Cohen's d of 0.25 are pretty big, at least compared to some of the other disorders. This is the group here. You see Sophie Townend, Graham Fairchild, Jules de Grey, Moji Agajani, Marlena Staginis, Stefan de Brito, and Charlotte Cecil in Amsterdam a couple of weeks ago. And just look at this. It's very interesting. What do you think about these patterns? So the two primarily externalizing disorders on the left, ADHD and conduct disorder, show very profound surface area reductions versus typically developing kids, whereas the two internalizing disorders on the right, anxiety and major depression, the effects are milder. They still affect surface area, but there were no internalizing specific effects. This is being presented by Sophie Townend on I think it's the MDD call recently. And this also, looking into the same question, is work by Willem Bruin and the anxiety group, trying to use machine learning to identify markers that are linked to anxiety. Is one of the more difficult disorders to study, but are linked with anxiety using machine learning. This appearing in Nature Mental Health. Very, very, very nice paper. Just to sort of digest what's been coming through, you all know there's an ENIGMA T-shirt, and it has all of these brain maps on. And someone can say, well, what are the general patterns that you see if you look at the different disorders? So Kuldeep Kumar and Sebastian Jacquemont have been looking at all of the disorders that ENIGMA has been studying, and they made a couple of general observations. They found that the disorders that emerge very early in embryonic life, so CNVs, genetic deletions and duplications, affect ontogenesis of the brain, they tend to affect the surface area of the brain more prominently. The neurodevelopmental disorders in the middle that arise in childhood and adolescence tend to affect surface area in some cases more prominently than cortical thickness. And then the disorders that emerge in adulthood, have an adult age of onset, tend to affect cortical thickness more than they affect surface area, with some qualifications relating to whether the person is medicated or not. So that's interesting. So they have this set of principles about the age of onset and whether or not the surface area or thickness are affected. They also noted that early onset disorders have excessive deficits in primary sensory motor cortex, but later on, the disorders tend to have a more transmodal association cortex pattern. And as we saw, Sophie Townend and her colleagues found that there's a strong externalizing pattern and a weak internalizing pattern, depending on the disorder type. So let's take this a little bit further. A lot of the ENIGMA groups have been pooling data from different clinical working groups and asking what are the common and distinct patterns. So Zhipeng Cao and Hugh Garavan have a paper there on the top right saying that all of these disorders share a common pattern. I mean, they may be a general P factor that is explaining it, but then they have disease-specific effects which were subjected to PCA by Sophie Volk and Micah Hetwer. So he said, well, look at all the ENIGMA maps. We'll get rid of the main component of deficits. Let's look at the principal components along which these brains tend to differ. And so they have a very nice paper on axes of transdiagnostic covariance with some deficits occurring primarily in association cortices and others in primary unimodal cortex. Now, the reason I bring this up is the new working group, the ENIGMA Gradient Working Group, that attempts to make sense of the multivariate patterns of anomalies in a framework many of you will be familiar with it, that sort of separates unimodal and heteromodal cortical effects. It's a very, very nice integrated framework to understand the developmental sequence, aging, and to categorize some of these disorders. Another question is, do the brain deficits tend to be bigger in disorders that are less prevalent? So this is an idea that Rebecca Cooper, Maria Jalbrzykowski, and David Glan proposed. If you want to be part of this, contact Rebecca Cooper. She's at the Boston Children's Hospital working with David Glan. She's a postdoc there. So have a look at this. So all of the Cohen's d effects on the y-axis. Let's look at the map on the picture on the left. The more prevalent disorders tend to have weaker effects. I mean, maybe major depression is an example of a very prevalent disorder with very, very weak effects. But then other things on the far left, you know, maybe neurogenetic syndromes are not very prevalent. So it's kind of interesting. Is this biologically based, or are the sample sizes of the studies a bit biased? I mean, the very interesting questions. If you want to be involved, have a chat with them. Very interesting question. And if you just want to put your working groups maps in the ENIGMA Toolbox, have a chat with Sophia. So just to remind everyone, Sarah Riviere and Boris Bernhardt have a toolbox that you can go and look at. And it has not just ENIGMA disease maps, but all sorts of maps of receptor distribution, brain connectivity, even cytoarchitecture, the Brodmann maps and so forth. So you can ask if the pattern you're seeing has any relationship to any of these other modalities of neuroscientific data. It's extremely interesting. And just as a reminder that this can be informative, Bratislav Misik and Justine Hansen in Montreal looked at the ENIGMA disorders and whether or not certain receptor patterns or connectivity patterns had a preferential involvement in certain disorders. Really fascinating way to look at this. The software Neuromaps has many of these things in. So if you want to put your disorder working groups maps in these softwares, I either chat with Sophia or the software developers, and that will make it easier to do these cross-modality studies. Well, one thing that made me sort of sit up and think, so Leonardo Cardoso Saraiva from New York and also Brazil. So, sorry, we couldn't find a picture of you, so we put a picture of Leonardo da Vinci. Sorry about that. But Isaac Sibelius in Cambridge, in England, they did the following. So they computed all sorts of cortical features. So we normally look at thickness and surface area, but they also looked at the surface curvature and gyrification and all sorts of other things. And I sort of fell out of my chair because a lot of the maps of case control differences showed bigger effects for the features we don't normally look at. So we'd better look at them. Now, just to give you an example, in Park at the Seoul National University Hospital in Korea, and the OCD working group have found that gyrification index, for example, is a good marker associated with OCD. So if you'd like to calculate these other measures too, have a chat with Leo and with Isaac, and they'll help you. I think that might be a nice direction. Some working group specific findings. So Isbrand van der Werf, Connor, Max, Ava, Emil have been running a lot of Parkinson's disease studies. Congrats to Emil for just getting his PhD. I heard you just graduated with your PhD, so congrats. Have a look at these. So there's four major papers mapping atrophy progression in the cortex, which is on the left, atrophy progression in the subcortical structures using Boris Gutmann's shape mapping software, atrophy progression in the cerebellum, which has been charted using Ian Harding and Beck Caresti's Acapulco software, and last of all, the microstructural anomalies as seen with DTI in a paper led by Connor Owens-Walton. Now, it's very interesting. We'll zoom in a little bit. So this looks like a tauopathy. I mean, which it isn't. It's a nucleopathy, but it looks like there's a progressive sequence in the cortex. But many of these maps in early PD were showing what we might describe as better than normal, or the other side of normal. I'll tell you what I mean by this. So this paper looking at DTI across 17 cohorts by Connor and colleagues here, they found that in the Hoehn-Yahr phase 1, stage 1, the fractional isotropy, which is normally lower in disease, was in fact higher. The mean diffusivity, which is normally higher in disease, was in fact lower. There's a lot of interest in whether this is inflammatory or compensatory or something else. It's very intriguing finding. Max Landsma and Boris Gutmann led a great study staging PD using these maps. So this is a wonderful study. So let's say you have maps of each stage. There's something called ordinal regression. And so you say, well, for my patient, which map does it best, does their image best fit? And their ordinal classifier, is a very interesting approach, will give you a readout. And that could be very interesting for therapeutic evaluation, among other things. Now, this paper generated huge interest. So Jianfeng Feng and Yuchao Zhang, with Jess and Theo's schizophrenia working group, studied 41 international cohorts with 11,000 MRIs. And they use sustain, and it's a subtyping and staging algorithm. They found that, let's look at it in some detail. Oh, there's the paper being discussed. So there's a wonderful meeting in Serbia where this paper is being discussed rather intensively. And they ran an algorithm called sustain, which pulls out subtypes and also stages, and let's look at them, with a conclusion that the course of atrophy in schizophrenia was not just one pattern, maybe like Alzheimer's disease, that there were sort of two subtypes in there, one starting in the frontal cortex and one starting in the temporal lobe. A lot of interest in whether there's anything different at the neuropsych level or the treatment response level about these, but the first step is to find them. If you're curious how it works, I mean, you look at all the brain measures and see when they become abnormal. I mean, they cross a sort of certain Z-score threshold. And if you only have cross-sectional data, you can see if one has done that more often than others and put them in a sort of sorted order. And I mean, it's very interesting work. I'd encourage you to take a look at that. You can use this in other disorders. So OCD was looked at with sustain by Ying Zhao, Hailin Cui, and Di Chen, and also Valerie Voon. And they identify 3 subtypes of OCD, also with progressive and even remitting changes, is sort of interesting. And they're also using the same shape mapping analysis that Boris Gutmann's lab developed. This is work by Yang He-Im and Chris Ching, mapping surface morphology and bipolar disorder. It's extremely interesting work, refining the spatial detail in the maps. And there's also work here by Darren Kobia in the schizophrenia group looking at subcortical shape details. They found some very interesting sex differences and laterality to the deficits that are seen in schizophrenia. Some groups published their first papers or got their first results. So this is really exciting work from Perry Pascu and Kevin Black and Yin Jin and others working with the Tourette syndrome data. This is rather a large sample compared to any prior study, and you'll see about zero point 0.4 Cohen's d, which is very large for a developmental condition, in adults, primarily affecting the frontal cortex, which, given the motor issues in Tourette's, is potentially a good lead. And then in the pediatric group, more diffuse effects that were found. Laura Berner has a really intriguing results in bulimia nervosa. So in the eating disorder working group, you can see some people running a 5K on the right. I think that's Stefan Ehrlich's team in Dresden. He leads the anorexia component of the eating disorders working group, but the bulimia group led by Laura Berner found reduced nucleus accumbens volume, which is part of the reward system, as you know, and also reduced cortical surface area. Again, these surface area findings are very, very prevalent in these developmental conditions. So that's pretty exciting. That's been submitted to SOBP. This is cool. Kevin Hilbert and colleagues published on specific phobia. This is phobia type of anxiety disorder that relates to a particular stimulus, either blood draws or injections, or people might have a phobia of a particular animal, spider or something like this. And despite the subtlety of this disorder, they're finding robust and consistent structural differences. Very tiny, 0.06 Cohen's d, is about 6% of a standard deviation. Effect size is similar to MDD, but the system's biology is extremely interesting. Just a shout out to Chris Ching and Ole Andreassen's bipolar disorder group. There's been a very active line of work led by Thomas Hayek in Dalhousie University in Nova Scotia. Here's Julia Freyja-Picado presenting at ISPD, and also an award-winning paper by L'Oreal Dietz and Thomas Hayek, showing that at least some of the deficits that you will see in bipolar disorder are attributable to high BMI. And so now that BMI is treatable, at least in some people's opinion, using GLP-1 agonists, semaglutide and such like, that becomes a therapeutically interesting line of work. On the genetics of the psychiatric disorders, Nadine Parker and Ole Andreassen have shown that you can predict whether someone has bipolar disorder to some degree of accuracy using polygenic risk. If you add an imaging-based risk, which sort of adds up the brain metrics in proportion to how different they are in bipolar, it does even better. At AUC with imaging and genetics of 0.7 is not bad at all, particularly for bipolar disorder. The genetics is better than the imaging. I don't know if I'm disappointed about that, but it's extremely interesting, and congrats, Nadine, for this very, very nice work. Very good work by Shayi Shen and Heather Wally in Edinburgh have shown that people with high polygenic risk scores for major depression. Now, that's a very difficult disorder to do genetics in. The GWAS, I think, has 2 million people in it. But they found in 11 studies and 50,000 people scanned with MRI that there very definitely are reductions. I mean, the orbital frontal gyres, many of these systems are implicated in major depression quite prominently. The hippocampus certainly is. So they were finding people with a high polygenic risk for depression have a phenotype on the way to what you would find in patients. This is true in PD. So Alan Dagger and Nushin Abbasi, really exciting study. They looked at people early in life before they could have Parkinson's and found that elevated polygenic risk for Parkinson's disease was linked with two surprising things. One is a pattern of atrophy that looks a little like Parkinson's, at least it correlates with it to a 0.37 correlation. This is highly significant. I mean, you can look at the two effects at the top. There's the genetic effect, and in B, on the bottom right, there's the disease effect. They kind of look a little similar. They also found that people with high polygenic risk for Parkinson's have a larger cortical surface area, which was found in Katrina Grasby and Neda Jahanshad's GWAS, that the genomic loci that influenced cortical surface area were among the ones that influenced PD. So it's very interesting. Alan Dagger has a theory of a greater number of cortical columns in PD. If you read the paper at the bottom, it's a fascinating discussion of why risk for PD and PD might amplify your cortical surface area, which is not in the neurology textbooks. but do read it. It's very interesting. This work by Boris Bernhardt, Alexander Ngo, and the ENIGMA Epilepsy Group finds a similar thing. So if you have a very high polygenic risk for hippocampal sclerosis, your brain signatures deviate from typical in a pattern that resembles epilepsy. Now, you can look at those lines that I've drawn there. So this is from their new paper. The picture at the bottom left is the signature of people with high polygenic risk. They have that sort of temporoparietal area of anomaly. And then you can go and see it again in people with temporal lobe epilepsy with hippocampal sclerosis. So it's extremely interesting. Very, very interesting result. And it might make you think that polygenic risk should be mapped using gradients. So those of you that like gradients will think that these things should be plotted in a gradient framework to see if there's a predilection for certain types of cortex. Another secondary project, this is fun. So Mr. Ben Sinclair, who is in Australia, is using PET data for the first time in ENIGMA. We haven't had a study in ENIGMA using PET data to predict whether you should be having surgery and which region to resect. It's a very good use of deep learning. So that just looked very exciting. It's a project using AI. Now, a section on pipeline updates. So VBM, championed by Matthew Kempton and his team, was used to reveal the profile of gray matter atrophy in early onset psychosis, and also the gray matter deficits were linked with chlorpromazine equivalent dose in people taking medication for early onset psychosis. So not to be deterred by having done one of these, he did another one. Here's one by Shane Prakar in Matthew's group, and look at this. It's very interesting. So in bipolar disorder, they looked at 34 cohorts with the help of Chris Ching and Olen Andreassen's bipolar disorder working group. Look at all of that deficit in the splenium of the corpus callosum, which would not be the area that would be famous for deficits in bipolar. I mean, there it is. I mean, it's partly ventricular, but it's very interesting. And so Chris Ching and Matt Kempton won a grant on this. It was very interesting. And they're now running VBM in major depression, schizophrenia, OCD. Actually finished the OCD project, PTSD and ADHD. So if you want to have a go at VBM in the disorder that you're working on, do ask Matt. Sophia Frangu, Rian Guy, Shalila Haas, Nicole Stanford and colleagues published CentileBrain. It's an enormous reference model, statistical normative chart of brain morphometry. It has a vast number of users. So look at that, 3,000 people have used it, which is unbelievable. So do check it out. The paper's very good. Riang recently gave a talk in Chinese on this, which is remarkable, and in English. So very, very commendable and interesting to give talks in different languages about how to use it. But we've had great feedback. Some of the active work on the top right, this is work by Fei Niu and others working with Sophia Franco, show that you can use CentileBrain perfectly well in people of different ancestries, which is an interesting question that they've answered in the affirmative. Down there on the left is all sorts of combinations of using CentileBrain with and without using combat to harmonize your data first. I mean, they're studying whether or not you need to use combat first, and it's sort of interesting ongoing work. Willem Bruin is also contributing to it. And they also have a brain age model that's part of this, which you're welcome to try. And this is great. So Han Wenbi, Masoud Tamarsian, and their colleagues are looking if they can detect signatures of insomnia using normative modeling. That's a very exciting use of CentileBrain. And then moving on to brain age. So James Cole is leading a brain age working group for ENIGMA. And we asked him what he recommended. So huge work by Laura Hahn and Tim Hahn, where you can use a brain age model trained on the ENIGMA MDD international cohorts. It's very good indeed. But there are also some others. So soon you'll get a questionnaire to ask the clinical working groups, do you have any brain age projects? What methods do you use? Do you like them? It might be interesting to do a cross-disorder project. The three methods in the middle are ones that James Cole recommends. I mean, he's been working on this a long time, so there are methods he recommends if you have free surfer or T1 or T2 or different types of inputs. And there are many, many brain age projects, but most notably Laura Hans, because she's published 2 of them, and a couple looking at apparent brain aging and HIV by Rachel Katzel, Ron Cohen, Jonathan Ipser, others. And it's a very, very interesting topic of apparently accelerated aging in these disorders. DTI. So big thank you to Neda Jahanshad and Peter Kochanov for supervising and creating workflows for diffusion tensor image quantification over the last 15 years. 25 working groups have published with them, and do have a look at papers that have been published if you're doing a DTI analysis. The newest one, this is really cool. So Caitlin Lloyd and Joanna Steinglass and others working with the anorexia group led by Stefan Ehrlich have found really strong effect sizes. So in anorexia, the structural metrics are about 1 standard deviation away, or a Cohen's d of -1 from the typically developing folks. Similar things are seen for DTI, at least in the corona radiata. And Caitlin has found that free water correction is actually very useful. We may want to integrate that step into the analytic protocols to see if it helps. Social anxiety disorder in work by Aline Roloffs at University of Leiden has subtle but consistent FA deficits that are more prominent. I'm going to get this wrong. It's more prominent in women than men. And that's a very cool study, very highly powered. Talia Neer, working with the HIV working group, has been finding microstructural differences associated with CD4 T cell counts, and even with viral load, which is a difficult association to detect in HIV across many cohorts worldwide. It's exciting work. And then finally, Jok Shah and colleagues, Rachel Marsh and Blair Simpson, have been looking at DTI in conjunction with machine learning to find that these are quite powerful predictors of OCD. Finding the internal capsule and the posterior thalamus, which is already implicated structurally, contributing to OCD classification. Alyssa Zhu, working with Neda Jahanshad and colleagues, have this brilliant framework for integrating multi-cohort DTI. So if you are doing a multi-site DTI study, use eHarmonize. The data on the left, which is FA across the lifespan, is completely chaotic. And if you use eHarmonize, it will tidy it up. Very similar work that's been done by Dylan Sun on structural data, comparing different versions of combat and showing that the better alignment can be achieved. Next year, we're beginning to roll out some tools for tractometry. So this work by Bram Chandio in our lab, Wendy Fang and Iyad Bagari in Neda Jahanshad's lab, is trying to refine the spatial detail provided from DTI. Just to sort of explain it, this is Bundle Analytics. It's a toolkit in DiPi, also co-written by Eleftherios Gerifalidis. And you can essentially, with these methods, this is one called META, or Medial Tractography Analysis, led by Iyad Bagari in Neda Jahanshad's lab. It will give you a long-tracked index of the association between microstructure and clinical outcome measures. So this has been used in a genetic context. Very nice work here by Iyad Bagari, showing that different sectors of tracks have not only high heritability, but there's genetic modules. And so the genetic correlation persists over a short range. And then there might be another sector of the track with different genetic determinations. Very interesting idea. Bram Chandio and Wendy Feng have been showing that amyloid and tau influence white matter microstructures. It's sort of interesting to maybe avoid a PET scan if you have Alzheimer's disease. And then Leila Nabulsi and Connor Owens-Walton in bipolar disorder and Parkinson's disease have shown that there's regional anatomic localization of those disorders in terms of their microstructural effects. It's a nice result on the upper left, showing frontal limbic deficits in bipolar, which are exactly what you would expect. There's a DTI GWAP. So if you have genetic data and DTI, you can join in. There's a cast of experts shown at the top, Barbara Maltz, Gabrielle Blockland, and others, Neda and Barbara Franke, who are leading this. So do join in with this. That's going to be a really, really exciting study. And if you have at least 100 people with DTI, do contact them, and they'll help you out to do the genetic association. There's some very nice new tools. Neda Jahanshah's lab has been creating software to view tracked specific effects. Very, very, very nice software. And also SMAC, which is a new algorithm authored by Shruti Gadawar in Neda's lab, was used by Ravi Bhatt, who got his PhD in the last week, to identify a large range of genomic loci that affect corpus callosum microstructures. It's a fascinating paper that you can see as a preprint that's been aided by Shruti's SMAC software. So if you want to extract the corpus callosum in your data, have a chat with Neda and Shruti, and they'll be able to give you a hand. White matter lesion quantification. So this comes up in aging, certainly in stroke, but to some degree in Parkinson's. And there's a great pipeline to look at it. So if you have T1 and flare, Sarah Al-Bachari from London, formerly of Lancaster University, has a pipeline that you can try. It's very good. Also in stroke, I mean, there's a great pipeline that Stuti Chakraborty and Sukli Liu have created. If you have an interest in perivascular space, it's implicated in all sorts of illnesses, they will give you a hand to measure these metrics. Many groups now run cerebellar analysis, thanks to Beck Carestes and Ian Harding. There's 5 papers published, lots underway. If you look at these results, here's Ashley Huggins' paper on PTSD with a regional deficit in the cerebellum. Finn Kelleher and Emily Dennis have found different degrees of brain injury, from mild to moderate to severe, have different degrees of cerebellar atrophies. Extremely interesting, and they also depend on the time since the injury. So these are pretty exciting papers. If you want to use Acapulco, have a chat with Ian and Beck, and they'll help you to do it. There's also a spinal cord toolbox. So those of you who think that MRI just chops off the spinal cord, you probably have a lot of scans like the ones in the picture that have at least some cervical vertebrae, C1, C2, with spinal cord. And Marcondes França and his colleagues have written a pipeline. Tiago Rezendi was the lead software author that will measure that. It's extremely interesting in spinocerebellar ataxia for obvious reasons, but it's been tried in Parkinson's. So this actually very hot off the press. The stages of Parkinson's disease seem to be reflected in the mean central surface area of the first 4 cervical spinal cord segments. Now, what's fascinating about this is that the reliability of these measures is very high. It's much higher than brain measures. And so these are sort of an unexpected bonus if you're scanning the spinal cord as part of your imaging. This software toolbox developed by Dylan Sun might be useful to all of you. So the image-based meta and mega-analysis toolbox, or IBMMA, will let you do voxel-based meta-analysis of signals either in images or on the cortex or in tables. Have a chat with Dylan. I mean, I know, Dylan, you created this as part of your work in PTSD, but it's phenomenally useful. So do have a chat with Dylan if you want to do image-based meta-analysis, which many of us do. Johanna Bayer and Philip Sayman wrote a masterwork on sight correction in imaging. It's absolutely wonderful. Everyone should read it. And they are now doing a project across multiple working groups with voxel-based morphometry to see what are the best harmonization steps you should take if you have voxel-based data. So have a chat with them if you're interested. MEG, so Alison Nugent, Anna Namist, and Jeff Trout have been creating a resting state MEG pipeline that they are using to study spatiotemporal patterns of brain oscillatory activity and relate them to age, sex, and genetics. If you have any MEG data, you can have a chat with them. They're quite brilliant, and they have a good protocol to process that data. We're soon to wrap up. We'll be done in about 5 more minutes. So there are some new groups. So the neuromodulation group, led by Taylor Kuhn and his colleagues, have a letter to the editor. It's a very nice overview of how these methods work. So if you're interested in these methods but don't really know how they work, do read it. It's extremely interesting. And they have a number of projects looking at predicting transcranial magnetic stimulation outcomes in major depression based on baseline brain imaging. Same thing in adolescents with depression, led by Danny Pine, Andrei Zugman, and Anderson Winkler. And a number of others. That one of those very exciting Tim Hahn's project uses control theory. How does neuromodulation parameters relate to the illness, and how could you optimally select them using control theory? Very, very interesting. David Mailer and Emily Haug and David Linden have been studying how to integrate neurofeedback data sets. If you want to join, you can reach out to them. It's extremely interesting. And then also Lauren Salmon and Megan Herting, JC Chen, and Teresa Monreal, who's the project manager, it's a funded working group to look at ENIGMA environment, which will get you environmental variables through geocoding. Now, a lot of people 2 weeks ago in Amsterdam had an epiphany. So when Lauren presented this, I know Kevin Black and others said, well, that's much easier than I thought, because the privacy issues with geocoding a patient's data, I mean, maybe there would be concerns about releasing their home address or anything about them, but it's done in a very clever way, which Lauren would explain, where the data is sent to a clearinghouse, and they just send you back the environmental data. I mean, things like environmental pollution, urban density, access to healthcare, I mean, all sorts of social environmental measures. And then they throw the geocoding data away, and so nobody will ever have access to it. So groups, particularly in Western Europe, where GDPR is like a sword of Damocles over your head, you will be able to do this. It's not an issue. So do have a chat with them. This is how it works. It's rather remarkable. So do have a look at how the exposure outputs can be returned to you and your cohort without compromising the privacy of the people who've been scanned. It's very interesting. There's great work by Emily Dennis and colleagues on neuropsych data harmonization. So let's say there's a wonderful paper on the left looking at verbal fluency, verbal learning, all sorts of questionnaires assess that, including ones in different languages. And they created a conversion approach to translate scores assessed on one test to scores on the other. Now, there's an entire branch of neuropsychiatry, which many of you are experts in, but a very clever way to do it on the right is to use BERT. So BERT is an AI transformer, which will read in questions in your symptom inventory questionnaire and find questions in a second inventory that are semantically similar. Now, I don't hope to understand how this works, but have a look at Eamon Kennedy and Emily Dennis's paper on that. I mean, that's attracted huge interest, because if you have symptom assessments that partially overlap from different sites, this will give you a principled method to interconvert one scale to another. And you don't have to take their word for it. I mean, you can check whether it does it in people that were assessed with both scales. It's pretty good. So that's worth taking a look. Another theme in ENIGMA is understanding medication effects. So this has been championed in a lot of groups. We mentioned the Neuromod group, CBT response being predicted by the OCD working group, and then a major effort on neuroscience-based nomenclature that's been led by Chris Ching and colleagues in National University of Ireland in Galway, Sinead King, who's now in London, and Colin MacDonald and Dara Cannon. Now, what they've been doing with Joaquim Radoa and colleagues in Barcelona is categorizing psychiatric medication, in a way that the ECNP recommends according to mode of action. So just look at this. Well, a different study first. This is predicting CBT response from first structural data, which seems to work, and then from fMRI data, which does not seem to work. Lawrence van der Mortel did a very good job of this, but it does not seem to be predictable from functional data. But then going back to this business of harmonizing medications, if you have a long litany of prescribed medications at different times, have a chat with Chris and Colm and Sinead King on how to group this in a way that will simplify the modeling of medication. And so just to show you a result, if you have bipolar disorder, you'll remember the ENIGMA map shows some deficits in hippocampus and frontolimbic areas. But then if you split the bipolar cohorts into those taking lithium, anti-epileptics, antipsychotics, this again is work by Colin McDonald and Sinead King working with the bipolar working group, you get a very different picture. I mean, lithium is a salt, and maybe it's hydrating the brain, but you'll see hypertrophy. And maybe there's some confounding with clinical severity, and this is not randomized. But in any case, this is worth being aware that different medication strata in the cohort will have quite different biosignatures. Nice work by Leila Nabalsi and Dara Cannon linking the use of SSRIs with actually connectivity metrics across many sites in ENIGMA BD. Last of all, but not least, there are ENIGMA working groups that have just begun. This is one on diabetes, led by Shan Wu at USC and Tamara Hershey at WashU. If you have any interest at all in diabetes, or you collected cardiometabolic information from people you've scanned, give them a shout. There's a call with lots of people already taking part. It's going to be a very interesting study. This is a study of developmental coordination disorders, very common, often comorbid with ADHD, led by Christian Hyde and Ian Fulcher at Deakin University in Australia. Christian traveled 10,000 miles to come to Amsterdam 2 weeks ago to meet everyone else. So thanks, Christian, for coming. And they're a very valuable new addition to the neurodevelopmental working groups. This is sort of interesting. So Christos Pliatsikas and many people who are very famous in the area of language, Simon Fisher in particular, are interested in whether you've collected data on multilingualism or anything about language, with it in mind that people who are multilingual may have a number of unique characteristics, including maybe better aging or better resilience to different conditions. So that's a cool one to reach out to them for. The meditation working group, Tony King, Sampras Kanasan, and Jess Turner, published their first report. There it is on the left. Do read it. It's absolutely fascinating how different meditation and contemplative practices not just beneficial, but also very valuable in the treatment of chronic pain. I mean, a number of disorders where other treatments have failed. You can read a little bit about what they're doing. It's one of the most chill working groups, which will not surprise you at all. So anyway, it's a good group of people to work with. And this group, led by, I won't single anyone out in particular. Nicole Peterson is the leader. But look at the cast of well-known researchers there in women's health and endocrine medicine. Karina Heller and Claudia Barth led this. It's a pitch for the neuroendocrine working group in Nature Mental Health. They're very interested in looking at hormones throughout life, either puberty or menopause or exogenous hormones such as contraceptive use, and how they influence brain changes throughout life. And they already have received 2,000 images. So if you've been collecting any steroidal or endocrine data, or for that matter pubertal status or menopause, andropause status, have a chat with them. This group is broken up into groups that study different parts of the lifespan. So you'll find a lot of colleagues that will be able to work with you on those subjects. Now, this is amazing. I'm not even allowed to tell you the results of this because Jan Kide and Sylvia and Neda told me not to. They have results that show a signature in people with chronic pain that I thought was extraordinary. We're not allowed to tell you them yet. But in different types of pain, migraine, which is neuropathic, or nociceptive pain that arises from a peripheral injury, they've been mapping biosignatures, and so do look out for that. I will not spill the beans on their results. Valentina Lorenzetti asked if we could start a group on experimental cannabinoids. So this is not users of cannabis as abnormal or illicit drug use, but it's as a treatment. Many of you know that cannabinoids are therapeutic as well. So she's interested in other groups that have been looking at cannabinoids and the parameters of administration. and how they're linked with brain and behavior and treatment effects. So do have a chat with Dr. Lorenzetti. And there's finally this group by Gina Hense and Henrik Walter at Charité in Berlin is looking at stress and resilience. And so even task-based fMRI responses to stressors and how they both impact the brain and general health. So do have a chat with Gina and Henrik if you're interested in that subject. Jacqueline Stevens asked if anyone has fNIRS, which I should know what it is, but if you don't know what it is, there's a definition of it there. It's a modality to assess brain function that's very useful in brain injured populations or other clinical populations. So if you have that type of data, give Jacqueline a shout and they'll start a working group on it. And then last of all, there's been a number of workshops. So let's have a look at these. This is Sophia Franco, Shalala Haas, Rian Gies, Translational Neuroimaging Program. These tutorials have been wonderful. One of them taught me about resting state, which I really enjoyed. And you know, you can learn to use CentileBrain in Chinese or English. But there's a lot of lectures that are on YouTube that you can look at. And if you have new students, it's a great resource. Thanks, Riang and Shalala, for doing that. Masood Tamasian has a series of sleep webinars. These are world-renowned experts in sleep talking about different aspects of the glymphatic system, sleep disturbances, and how to study these different things. So thanks, Masood, and all the people who've presented. Enigma U is a phenomenal international effort led by Emily Dennis and her colleagues in Utah. She's asked if anyone wants to give a short lecture. This is a program for students in many parts of the world that don't have access to neuroscience education. It's been phenomenally successful. If you'd like to give a lecture on the disorder you study, or its risk factors, or its comorbidities, approximately at the level of an entering undergrad class, but only 15 minutes. I mean, all of you that have contributed these videos, if you want to look at them, there's a Google Drive there that you can listen to these. They're phenomenally helpful for new students. So if you have a topic that you are willing to talk about for 15 minutes, I mean, these have been extraordinary. And have a chat with Emily about how to take part. This workshop, again led by Valentina Lorenzetti, was looking at addiction and particularly sex differences in addiction. And you can see Valentina and her colleagues. Sophia did a wonderful job recording all this and creating a YouTube channel that you could listen to hear about that. And then there's a very important initiative. Carrie Osapenko, who works in the brain injury working group in a subgroup focused on intimate partner violence. They have, I mean, I would not be able to summarize it. They have a wonderful program for support of people that have experienced domestic violence. I mean, not only the science, but also the practical aspects of brain injury in that context. And Dr. Osapenko has actually received a number of awards for this, including from the National Neurotrauma Society. So congrats to Dr. Osapenko. And there they are. There she is in the middle, and lots of brain injury experts. And this is the network, the knowledge exchange network for IPV. Last of all, although I keep saying last of all, there's a booklet. And so Gaun Kim, who's a student working in Kathy Lawrence's lab, and Sophia made this fantastic brochure of all of the neurodevelopmental working groups and the allied methods working groups. So do take a look at it. It's a very good summary of what they're up to. And if you'd like a booklet, you can ask Sophia if she give you a hand in putting one together. There are some for patients as well. So those of you that are working with patients and caregivers, the bipolar working group made a booklet for patients and families. And it has a little bit of research in it, but also more practical information. So that's a nice and useful outreach effort. Oh, if you want to work in Birmingham, so I was there 2 weeks ago visiting Stefan De Brito. They're hiring 125 professors, including some in our field. So if you'd like to go and work in Birmingham, it's a beautiful university. It's an extremely nice place to work. Do have a chat with Dr. De Brito about applying. Maybe I'll be repatriated and go and work in Birmingham. Okay, next year. So that's the end of this year. Next year there'll be a meeting in Toronto at SOBP. We heard yesterday some interesting news. OHBM, many of you submitted abstracts to in the last couple of days. An email came in yesterday saying, would you like to give the opening plenary lecture of OHBM? It's called the Telerex Lecture to 3,000 people and talk about Enigma. So thank you to whoever thought that was a good idea. We'll do our best. But on a more serious note, I mean, Enigma partly began in Brisbane, where the meeting is. So Sarah Medlin, Nick Martin, not just the Australian group, but Barbara Franke, Neda, David Glan, many others were instrumental in Enigma forming 15 years ago. So it will actually be rather nice to have a celebration of Enigma at the beginning of OHBM. That will be the first talk of OHBM on the first day at about 6 o'clock on the Sunday. I think it's on the 24th. The other meetings that people go to, so do let us know which conference you go to. There's even a little form that you can fill in, and I mean it will help you find other people that might be going there as well, and we can plan get-togethers of various kinds. Oh, two journal special issues. Ali Safet Gönül and his colleagues Mesut Çetin in Turkey are inviting Enigma researchers to submit papers on Enigma studies. This is a very nice journal that is having a special issue. And there are going to be 10 submissions that you don't have to pay page charges for. So do have a chat with Dr. Gönül if you want to submit anything. And Neda Jahanshad also is organizing a special issue on imaging genetics. And so if you do anything with genetics and imaging, send your papers there or have a chat with her on whether that's a good venue. Big thank you to Andre Alleman. He has co-led for 6 years the Schizotypy Working Group with Gemma Modinos. He's now going to be Dean of Faculty in Psychology and Neuroscience at Maastricht, so he's stepping down. Huge thanks for your leadership, Andre, and good luck with the next job, which sounds even more difficult. And Matthias Kirchner from in Switzerland is going to very ably take over the role of co-chair of the Schizotypy Working Group. Thanks, Matthias, for doing that. Oh, this is our last announcement. If you're not getting any email, which sounds good actually, but if you're not getting it from us about this call, for example, tell Sophia. And I mean, people that are lead analysts in your group and your members. I mean, if you're not getting emails such as this one and would like to get them, we can figure out why you're not getting them. So, just in closing, thanks for a great scientific year. I mean, who knew when we began on this odyssey that there would be such a landscape of activity to review? I mean, we can't even get through it all in 80 minutes. But I just want to thank each and every one of you. I mean, you are making enormous progress in a field that's very difficult, and by working together, we can achieve discoveries that no one of us could achieve on our own. I want to thank all of you for your leadership and vision. And if you're heading out for the holiday, have a safe journey. If you're a medical person who doesn't get a holiday, thanks for your service. And we'll look forward to seeing you in 2025.